Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the complement system. In the last video, I've told you about one of the important complement system pathway, that was the classical complement pathway. And in this particular video, I want to focus on another important pathway of the complement system, which is known as the alternative complement pathway. This was an overall view of the complement system, and I've told you that there is a classical pathway and then there is an alternative pathway, and we had a detailed discussion on the classical pathway in the last video. In the last video, we had a detailed discussion on the nomenclature of the complement protein. Let me summarize that for you. And I've told you that the complement proteins, they are designated by a capital letter C followed by a number from 1 to 9. For example, the C1, C2, C3 till the C9. And these numbers, they are actually showing the order of the discovery of this complement protein. Then I've told you that the complement proteins, they are made in their inactive form, so they are there in the serum in their inactive form unless their activation is needed. And when you are interested in the activation of this complement protein, you are going for the cleavage of this complement protein, and the cleavage is resulting in peptide fragments, which are denoted by adding a lower suffix a and small b. Then I've told you that in most of the cases, the smaller fragment resulting from this particular cleavage of the complement protein, that is designated as small a, and the larger fragment is designated as small b. C2 is an exception where these particular rules, they are the other way around. Then I've told you that why we call the complement system as a complement system, because that is part of the like humoral or innate immunity. But the reason we call this as the complement system was that it enhances or it complements the ability of the antibodies and the phagocytic cell. So it is complementing the immune system. We are calling this as a complement system. And then I've told you that this complement system is very important to clear the microbes and damaged cells from an organism, and they contain a number of small inactive liver synthesized protein precursors circulating in the blood. And if you look at the number of proteins and the protein fragments that are included in the complement system, their number is about 50. Then I've told you that there are three biochemical pathways for the complement system. One was the classical complement pathway. We had a detailed discussion on that in the last video. The second one is the alternative complement pathway, which we are going to see in this particular video. And the lectin pathway, that will be the focus of discussion in the next video. So let us start about the alternative complement pathway, how this works. If you remember, I've told you that when you talk about the classical complement pathway, you need an antigen antibody complexes. Now, in case of the alternative complement pathway, you are not going to need any antigen antibody complexes. So, what you need for the activation of an alternative complement pathway? A lot of things that can activate the alternative complement pathway. For example, the teichoic acids present on the gram-positive bacteria, that can be one of the inducers of the alternative complement pathway. Then on the gram-negative bacteria, these lipopolysaccharides, they can be another activator of the alternative complement pathway. When you talk about the fungus or the yeast cells, so Zymosan, which is actually a fungal and the yeast wall cell component, that is another component of that is another activator of the alternative complement pathway. So you can activate this in many ways. When you talk about the alternative complement pathway, four proteins, they are very important. The C3 protein, the factor B, the factor D, and the properdin. And these three factors, the factor B, the factor D, and the properdin, you are not going to see them in the classical pathway. So they would be specific for the alternative complement pathway. So how this pathway starts? Uh, when you talk about the C3 proteins, so a small fraction of the C3 that undergoes spontaneous hydrolysis in the absence of the pathogens. So what you are going to see is that if this is the C3, So a slow spontaneous hydrolysis, that is going to give you the C3a, the smaller fragment, and that is going to give you an activated C3b. So this particular hydrolysis of the C3 into the C3a and the C3b, this is spontaneous hydrolysis. And this particular C3b is not going to interact with the pathogen surface. It is not going to interact with the teichoic acid or the lipopolysaccharide or the zymosan of the fungal or the yeast cell, but this is going to activate the complement, the alternative complement pathway. So let us discuss about the formation of the C3 convertase. We have already seen how the C3 convertase that is made in the classical pathway. So how this is formed in the alternative pathway. Let us see this. So when you are going to cleave the C3 spontaneously into the C3a and the C3b, so I'm going to represent this C3b by this red ball over here. So this is represented as C3 H2O, and it is also known as C3b H2O, because we have cleaved it by the process of the hydrolysis. So you can expect a water molecule over here. So what this C3b do is it is going to bind to another factor, which is known as the factor B, which is actually a plasma protein. So the C3b, when it is going to bind to the factor B, you are going to get a complex of the C3b and the factor B. Now this particular complex of the C3b and the factor B, that is stabilized by a magnesium ion. So magnesium ion is going to give the stability to the complex of the C3b and the factor B. Now when the C3b it binds to the factor B, it is going to make conformational changes in the factor B, and these conformational changes actually allow the binding of another protein or another factor, which is known as the factor D, to the factor B. So the factor D that is going to bind to the factor B, and this binding actually leads to the cleavage of the factor B into the BB and the BA. So this is going to be the larger fragment, and this is going to be the smaller fragment. So you have cleaved the factor B into the BB, and you have cleaved that into the BE. Now, what happens is that this particular C3 convertase, this C3b and the BB, as you can see over here, we are going to interact the factor B with the factor D in the presence of the C3b. So when the factor B that is cleaved, this factor BB is still attached to this particular CB over C3b over here. So this C3b BB, this particular C3 convertase, what it do is that it is an unstable complex. So now you have got a complex of this C3b and the BB, but this particular complex is unstable. So this particular C3 convertase needs stabilization to be effective. By that I mean that if the C3 convertase have to act on the C3 to cleave that into the C3a and the C3b, you need a stabilized C3 convertase. And this particular complex of the C3b and BB is not a stabilized one, it is an unstable one. So how are you going to stabilize this? There is a regulatory protein which is known as the properdin, and this properdin is actually going to bind to this C3 convertase. So you are going to actually get a complex of the C3b, the BB, and the properdin, and this particular complex of these three stuff is going to give you a stabilized C3 convertase. So you can actually see this stabilized C3 convertase in the form of the C3b, C3b, BB, and P. And this particular complex, and this stabilized C3 convertase, is now able to act on the C3, and when it acts on the C3, it is going to cleave that into the C3a and into the C3b, as I have represented the C3b that was spontaneously generated by this particular image. So again, I am representing the C3b by this particular image. Now, this particular C3b, which have been produced by the C3 convertase and not spontaneously produced, this is actually the C3b which is going to bind to the surface of the antigen. For example, it is going to bind to the teichoic acid of the lipopolysaccharide or the zymosan, for example. Now, when the C3b that is binding to the surface of the antigen, now you are going to make the C5 convertase. So, how are you going to make this particular C5 convertase? As you have already seen in the previous slide, that the C3b, BB, and the P that was making a stabilized C3 convertase. Now you have cleaved the C3 utilizing the C3 convertase, and you are getting this C3b. So this particular complex, this C3b, this C3b is going to be produced spontaneously. Then that was interacting with the BB. Now this particular C3b has been produced by the C3 convertase. So when these three combine with each other, you are going to get the C5 convertase. And when you get this C5 convertase, as we have discussed in the last video, it is going to be responsible for the breakdown of the C5 into the C5a and the C5b. So do not get confused these two C3b. This C3b is the one that has been spontaneously produced, and this C3b was the one that was made by the C3 convertase. So this particular whole complex is the C5 convertase, giving you C5a, C5b by the cleavage of the C5. Now, in the next step, you are going to make the membrane attack complex, because this is actually the membrane attack complex that is going to neutralize the pathogen, whether that is the gram positive one or that is the gram negative one, for example. Now, this C5b, as you can see over here, the larger fragment of the C5, that is now going to interact with the C6. So you are getting a complex of the C5b and the C6 now. Now, all of the complementation reactions, starting from the formation of this particular C3b that we got from the C3 convertase. So, starting from here, and all of the complementation reaction till the binding of the C6 to the C5b, they take place on the hydrophilic surface of the target membrane. So you are still on the surface of the pathogen. You have not yet inserted yourself, you have not yet inserted the complement proteins into the membrane of the pathogens. If you can see over here, the C5b, the C6, so you are still on the surface, but when the C7 complement protein, it combines with this particular complex of the C5b and the C6, you are going to see a transition from the membrane surface into the interior of the membrane of the pathogen. As you can see over here, so you are on the surface, but the addition of the C7 is going to insert this complement protein into the membrane. Another important thing is that when the C5b, C6, and the C7, they are going to provide a binding site to the C8, and again you are going to see a transition. So the complement protein C5b, C6, C7, and the C8, again you are going to move into the membrane. So you are going to go deep into the membrane of the pathogens now. Now, as you can see over here, when the C9 complement proteins, they combine with this C5b, C6, C7, C8, and the C9, so you are going to get a channel. You are going to see a pore in the membrane, and this particular pore is actually having a size of 70 to 100 angstrom in the target. Say for example, if this is the gram-positive bacteria, so now you have made a pore size of 70 to 100 angstrom. So you, after the C5b, C6, after this particular complex, when the C7, C8 combines with that, the C9 is then going to make this particular pore. Now, when the spore is found in the membranes of the pathogen, so what is the end result? Now, this particular pore formation is actually going to allow the ions and the small molecules to diffuse freely across the membrane, because now there is a pore. So the small molecules and the ions, they are going to diffuse freely across the membrane of the pathogen. Now, this is going to compromise the integrity of the membrane of the pathogen, because now they are not able to maintain the small ions and the molecules inside the cell, and that is because of the compromise in the integrity of the membrane pathogen. Now, when the bacteria, when the pathogen, they are going to lose essential electrolytes from the microbial cell, this is going to lead to the death of those particular cells. Now this is one phenomena, that the things they are going to be leaked from the pathogen cells. Secondly, the inflow of the extracellular fluids like the water, ions, and other small molecules into the microbial cell, that is going to, you can say, allow the microbial cell to not maintain its osmotic stability, and eventually they get the pathogen that get lysed and killed. So you are going to use two phenomena. The spore formation is allow the leakage of the important ions and the small molecules from the pathogen. Secondly, the inflow of the water and the other unwanted molecule, I would say, they are going to disturb the osmotic stability and eventually killing the pathogens. Now, you might be thinking that do these complement proteins of the alternative complement pathway, do they interact with the host cells? Like if you talk about the human beings, so do they interact with the host cells? And the answer is yes, they can interact. But there are sialic acid on the surface of the membrane on the surface of the mammalian cells. Like this is one of the this is the structure of the sialic acid, and the sialic acid is the most prevalent sugar found on the surface of the mammalian cell. Now, the sialic acid is actually working as a protective layer, and it prevents the binding of the complement protein, such as the C3b, to the cell surface. And when the C3b that is not allowed to bind to the surface of the mammalian cells, or you can say that is making the interaction of the C3b to the mammalian cell very poor, and thereby the complement activation cannot take place on the mammalian cells, and only the cells of the pathogen, they are degraded or they are killed. So if you have any question on the alternative complement pathway, you can ask me in the comment section, or you can ask me in the question section. We will continue the discussion in the next video.